Hi folks, welcome to Wildlife Cake and Cocktails. I'm your host, Yanni Torcola. Just a few quick notes. You can find the audio podcast on Apple, Spotify, Stitcher, Anchor, and more, and videos on our YouTube. Please like the channel and follow the podcast on our social media, and there's plenty more WCC on the way. Now, with everything that's going on in the world, obviously there's a lot happening with the Black Lives Matter movement, and here in Australia as well with the Indigenous Lives Matter movement. There's just far too many Indigenous deaths that have occurred in custody, not just here, but throughout the world, but definitely in this country as well. We urge you to check out some of the links below, including to Deadly Science, who promote the participation of Indigenous people through education and science here in Australia. They're a fantastic program. We recommend you check them out. And now, on to the show. Cheers. Hi folks, welcome to WCC Aperitifs. I've got a lovely gin and tonic, and I have a fantastic piece of banana cake here in front of me. And for a specific reason, in fact, we're going to be talking about polyploidy. And now we need a little bit of background here as well. So what is polyploidy, and why do I have a banana in front of me specifically? So polyploidy, or whole genome duplication, is a process by which the entire set of chromosomes of an organism gets doubled, tripled, quadrupled, or more. In most animals, including humans, that generally leads to some pretty erroneous genetic function. However, that's not always the case. Most animals are diploid, but a lot of plants are polyploids. 3N, 4N. So us diploids are what we call 2N. We have two copies of our chromosomes, one from each parent. Polyploids have more. So for example, in 4N tetraploids, you have both sets of chromosomes from a parent included into the offspring. While this is obviously typically disruptive, there's plenty of cases, such as in the breeding of hibiscuses, which can be, I believe, up to like 6N hexaploids. And there's obviously selection for agricultural purposes, such as my 3N triploid banana, which is seedless and edible because of this triploid condition. It doesn't reach the similar kind of reproductive heights as its natural seedless cousins do. So let's have a look at that here, actually. I've got a lovely picture here of a, well, this is our regular banana, right? And you can see not a lot of seeds there throughout the middle. And this is a wild type banana. And you can see the lack of flesh on the inside, much more seeds. Let's just have a quick look at here. So let's just imagine an organism with 3 sets of chromosomes. If it were haploid, it would just have 1, 2, 3, as you can see up here on the top left in the haploid N condition. In diploid, which is much more like us, except we have 23 pairs instead of just 3 here, but there is 2 copies. In triploid, like my banana, we get a 3N condition like this. And in tetraploid, 4N, this is, obviously 4 copies. And that's actually what we're going to be talking about. We're going to be talking about a tetraploid frog. So we will be discussing a fascinating paper, and I'm going to bring it up for you right now, if I can. There we go. So polyploidy breaks speciation barriers in Australian burrowing frogs, Neobatrachus. This is from Polina Novikova. You can find her on Twitter at Polina_Novik. Obviously a fantastic paper from PLOS Genetics, just recently out. You can find a link to that down below. So polyploidy. Obviously, there's different modes and varieties of polyploidy, such as order polyploidy, which is within species. So you have 2 parents which are of the same species, and they both pass down 2 chromosomes into an offspring. Allopolyploidy is a little bit different, where you get a hybrid of 2 closely related species, the zygote, or the offspring of 2 parents, 2 different species, that normally leads to a non-sexually reproductive hybrid, right? But that's not always the case. There's some rare exceptions, but fairly commonly in some amphibians, including the African clawed frog, which is a really important model organism for a lot of biology. So there's multiple independent diploid, sexually reproducing polyploid sister species, but the direct fitness advantages are a little bit unknown. And this includes in the Neobatrachus, a widespread burrowing frog genus in Australia. So the genus comprises 6 diploid species, which are 2N, and 3 tetraploid species, which are 4N. Fascinating. And they're all characterized by bisexual reproduction. So they're not, their reproduction hasn't been messed to the point that they're basically have to clone themselves the way a lot of polyploids do. They reproduce bisexually. In fact, they have distinct morphology and calls, even in some of these tetraploid species, which include Neobatrachus aquilonius, Cunipolari, and Sudele. So let us have a look at this paper here a little bit. Now, how did they investigate some of this? So what they used was an anchored hybrid enrichment approach for their sequencing, which we're really going to fly through a lot of this stuff. We're not going to have time to touch on everything that they did, because it's a fascinating paper. But yeah, we'd be here all night. All right, so we're just going to quickly go over the author summary here. So polyploidy, or whole genome duplication, is rare in animals, and usually the animals reproduce asexually. The Australian burrowing frogs of the genus Neobatrachus form an interesting exception amongst vertebrates with multiple independently originated autotetraploid sexual species. We generated population genomic data for 87 animals representing all 6 diploid and 3 tetraploid species of Neobatrachus. We show that while diploid Neobatrachus species seem to be isolated from each other, their sister tetraploid species experience substantial levels of gene flow and have wider distributions. Furthermore, we observe asymmetric gene flow from diploids to tetraploids. Based on our genomic and climate analysis, we suggest that such interspecific hybridization mediated by whole genome duplications rescues species diversity and allows tetraploids to more easily avoid climate-induced impacts of climate-induced habitat loss. So fascinating stuff here. Let's just have a look at this figure right here. So interesting stuff. On the left, what we have is 2 different trees. And these are, I believe, produced using the maximum likelihood method and a coalescent method in Astral. So Raxmore for the maximum likelihood, and the Astral coalescent method. And the dated species tree on the right is from Starbeast. Yeah, fascinating stuff. I guess some quick take-home points from this paper. Let us have a look here. There has been a rapid diversification in Neobatrachus, multiple independent origins of tetraploid species, which you can kind of see here on the left, right? Let's actually go down to this other figure here, where you can really see here in figure A, the bold species, Cunipolari, Sudele, aquilonius, they are the tetraploid species, right? So you can see that then they're quite separate. They're particularly Cunipolari here is closest to albipes rather than any of the other tetraploids. So it's not like there was one event where things became tetraploid, and then from there, the new species subsequently also came from that one tetraploid. No, these are independent events. Down here at the bottom, you can kind of see in B, you can see the rates of introgression between the different species, and a lot of those lines really seem to be going towards those dark, bold colored tetraploid species. And here in F, you can, as well, you can see the number of times a particular directional migration event happened, highest in Sudele to Cunipolari. So even amongst themselves, the tetraploids share a lot of genes, much more compared to the rest of the Neobatrachus. Fascinating stuff. So Interesting, right? We have these tetraploid Neobatrachus, and they share genes more effectively. They share genes more efficiently. They have this ability to maybe even pick up adaptive divergence from their diploid sister species, and that has allowed them to, by some of the species modeling and climate modeling, species distribution models and climate modeling that was done in this paper, it shows that adaptive genetic diversity has allowed the tetraploids to be more suitable throughout a changing climate, which might be something that we need to consider in the future. The tetraploids might be more able to deal with rapidly changing environments and future climate change as well. So obviously, this is something that should require some more work and some more monitoring. The authors suggest sequencing of whole genomes to look at kind of rates of introgression and genomic locations of introgression and all these other amazing things that can come out of this really kind of interesting model system for tetraploidy, or I guess autopolyploidy in a vertebrate. Fascinating stuff. Luckily as well, there's those emerging citizen science methods like Frog ID to maybe provide some of that information and monitoring in the future. Either way, fascinating paper. Thank you again to the authors. We'll put a link down below, and you can check out at Polina_Novik on Twitter if you want to follow the lead author here as well. Thanks again for the paper. We really enjoyed it, and there's much more wildlife cake and cocktails on the way soon. Cheers, guys.